Figeno, a figure generator for genomics. With Figeno, you can visualize various types of sequencing data, like ChIP-seq or ATAC-seq in bigwig format, high-C data, nanopore data with base modifications, and whole genome sequencing data with copy numbers and breakpoints. You can of course combine different tracks however you like, and you can display several regions simultaneously, which can for example be used to visualize chromatin interactions across a breakpoint. In this video, I'll show you how to install Figeno, how to use it, and all the tracks provided with Figeno. Figeno is a Python package, but don't let that scare you away because you don't need any knowledge of Python to use Figeno. In an environment with Python version at least 3.7, you can install Figeno by simply typing into the terminal pip install Figeno. This should work well on Linux and Mac, but Figeno has some dependencies which do not officially support Windows. To make it easier for Windows users, we directly provide Windows binaries for Figeno. If you head over to the Figeno GitHub repository and navigate to the release page, you can find the download link for the Figeno binaries either as a zip file or as an installer. Figeno has a graphical user interface, a command line interface, and a Python API. I'll start with the graphical interface since it's probably the easiest one to use. You can open the GUI by typing Figeno GUI in the terminal, or on Windows by starting the executable. This will open this page, which is where you can describe your figure. It's not as interactive as IGV, so you have to fully describe your figure and then click on Generate Figure before you can actually see it. The parameters are split into 5 categories. The first section is General, which is where you can select the layout and the reference genome. For most purposes, you'll want to keep the layout as horizontal, but there are others available, mainly intended for whole genome sequencing data, where you can for example use circular plots. Figeno supports any reference genome. HG19, HG38 and MM10 are already built in, so you don't need to provide any files if you're using one of those references. If you want to use another reference, you can select Custom, and you then need to provide the additional files. The format of these files is described in the Figeno documentation, and they can be simply downloaded from the UCSC table browser. Then, you need to provide the output file, which you can do by either directly typing the path to the file, or by using a file dialog. You can also change the resolution and the width of the figure. You then need to indicate which regions you want to display. You can either manually input the coordinates, or select a region around a gene. The next step is to add the tracks that you want to display. For example, if you have ChIP-seq data in bigwig format, you can add a bigwig track. When a required field is missing, it is highlighted in orange, so here we at least need to specify the path to our file. Once this is done, you can click on Generate Figure, and here is the result. In most cases, you'll also want to have a chromosomal axis and a genes track, which you can also add. You can reorder tracks by dragging the handle on the left and dropping the track where you want it to be located. With the buttons on the right, you can copy a track or delete it. A faster way to set up the 3 tracks would be to go to Open Template and select Bigwig, which is equivalent to adding the 3 tracks. You can highlight a region, for example here a peak, by adding a highlight and specifying the coordinates. At any time, you can save your current configuration in a JSON file. This config file can then be loaded again if you want to keep on editing your figure. The config file can be opened in a text editor, and in fact you don't need the graphical user interface. You can directly write the config file in the text editor, and then run Figeno make config.json to generate the figure. From the command line, you can use Figeno init to initialize the config file, which you then need to edit in text editor before running Figeno make. Finally, you can import Figeno as a Python module, write the configuration as a dictionary, and call Figeno make. We've already seen 3 types of tracks: chromosome axis, genes, and bigwig. In the first column, there are options which are common to all tracks. You can adjust the height of each track and the space between tracks. You can have a bounding box around each track, change the font size, write a label, which can be horizontal or vertical. For the chromosome axis, 3 styles are available. The default style shows ticks at fixed intervals, but there is also the ideogram style, which shows where the region lies within the chromosome. You can also use 2 axes with different styles at the same time. To avoid having the chromosome name written twice, you can disable it for the ideogram track. You can also put the chromosome axis at the top of the figure, and in that case you'll want to put the ticks above the line. The final style is called arrow, which will show an arrow with the start and end coordinates of the region. With this style, a color can be assigned to each region. You can change the color by clicking on the button, which opens such a panel. You can choose one of the predefined colors, or select the color using the Use Saturation Lightness, or manually enter the hexadecimal value of the color. This style is particularly well suited for displaying multiple regions. By the way, if the start coordinate is greater than the end, the region will be shown in inverse orientation. For a bigwig track, you can change the color and the number of pins. By default, the scale is automatic, but you can also set it to a custom value. You can also change the position of the scale. If you want to load multiple tracks, you can click on Open Files and select multiple bigwig files at the same time. Figeno also provides a tag for bed files to display specific regions, for example CPG islands. There is also a coverage tag to show the coverage for a BAM file, for example here RNA-seq. Next, the Hi-C tag lets you display Hi-C data in cool format. You can change the maximum distance between points where interactions will be shown, and if you untick Extend, only interactions where both partners are within the region will be shown, so some space will not be used. If there is a breakpoint linking 2 different regions, you can visualize the interactions across these regions. You can also disable these interactions across regions if you just want to show several regions side by side. With the alignment tags, you can visualize aligned reads from a BAM file. If you have nanopore data with MM and ML tags in the BAM file, you can display these base modifications. By default, methylated cytosines are colored in red and unmethylated in blue. But you can change these colors or even show other base modifications. Even if you export the figure to vector graphics, the base modifications will be rasterized, so you can still benefit from increasing the resolution. If the reads were phased and have an HP tag, you can also group the reads by haplotype, which here reveals haplotype-specific methylation, because one of the allele is translocated and activated by enhancer hijacking. You can choose to show the unphased reads or omit them. The vertical gap between reads is 30% by default, but you may want to reduce it in order to see methylation patterns more clearly. Finally, you can link split reads which are aligned to different regions to show structural rearrangements. For base modifications, you can also use the base mod frequency track. This can take as input BAM files with MM and ML tags, bed methyl files produced by modkits which have to be compressed and indexed, or simple TLSV files with columns for chromosome, position, and methylation percentage. If you have a BAM file, you can also split by haplotype, and you can potentially combine multiple files in the same track. If you want to show allele-specific methylation with nanopore data, you can start from the corresponding template, which already has an alignment track colored by methylation and split by haplotype, as well as the base mod frag track, so you just need to provide the path to the BAM files. The final data type that can be displayed with Figeno is whole genome sequencing data. Again, you can start from the corresponding template. There are 2 main tags, SV and copynumbers. The structural variants can be provided with a VCF or a TSV file. The format for the copynumber input files are described in the documentation, but basically you need a file to indicate the ratios in each pin and one to indicate the copynumber alterations, which are used to color the dots. If you omit the start and end coordinates, Figeno will assume that the region spans the whole chromosome, which is often what you want for whole genome sequencing data. Breakpoints within one chromosome are colored depending on the orientation of the breakpoints, and breakpoints leading to other regions are indicated by a line and the name of the other chromosome. By default, copynumber gains are shown in red and losses in blue, but this can be changed. You can of course show several chromosomes in the same figure. Figeno provides a symmetrical layout, which can work well when you show between 2 and 6 chromosomes in the same figure. There is also a circular layout. With this layout, each region has a color, and you may want to remove the vertical grid in order to make the figure clearer. This circular layout can display all chromosomes to summarize all structural rearrangements in a sample. For this, you can either click on Add All Chromosomes or select the circles template. So this was a quick overview of Figeno. If you want more details, you can have a look at the documentation. I hope you'll find Figeno useful, and if you use it, don't hesitate to provide feedback.